So thank you very much for this opportunity to speak on the role of the cerebellum in dystonia. My name is Dr. Anna Sidnitska, and I'm a child-brain clinical lecturer in medicine in London. Now, we've known for many years that the cerebellum is likely to be implicated in the pathophysiology of dystonia. Here's a paper from the 1960s in the New England Journal of Medicine by a neurosurgeon, Cooper, who was already an advocate for extending lesions in the thalamus more posteriorly so that we would purposefully interrupt the afferent fibers received from the cerebellum, and in documented cases there was dramatic improvement in the dystonic contractions in this patient with torticollis. We also know at a very basic level, despite a huge expansion in the number of genetic mutations that are thought to lead to dystonia, that these are all expressed within the cerebellum. And so this is a viable candidate for a pathophysiological driving mechanism. The number of publications investigating dystonia steadily increases over the years. And the cerebellum within this is reached an almost exponential level. Over the last decade, there has been a huge expansion in the number of papers exploring this topic. The specific question that I wanted to examine in this talk is examining human studies in patients with isolated dystonia and attempting to look at core cerebellar computations, adaptation and idling conditioning, and then discussing where we may see future research questions go and the strategies that we can employ to explore these questions. So to this audience, I do not need to explain the phenomena of motor adaptation, which can be defined as the recalibration of well-trained movements such as locomotion, eye or reaching movements to changes in the environment. This is a form of error-based learning where we believe that the brain computes an error teaching signal between the actual consequences of the motor action and the anticipated motor actions. And we believe that the cerebellum is involved integrally in many of the different steps involved in this computation. Now, back in 2014, I started exploring adaptation in isolated dystonia, as there was a real growth in this idea that the cerebellum was involved in dystonia, and could we try and tap into causative mechanisms? I used a very classical paradigm. This was using a purpose-built robot at UCL and Imperial, and we had a simple reaching task to one of 4 targets. And then we used both the visio-motor perturbation, where we rotated the visual feedback by 30 degrees, and also a force field perturbation, more reliant on the proprioceptive feedback to induce a similar magnitude of learning error. And this was all work in collaboration with Professor Joe Galea, and we were both struck by how amazingly intact this phenomena appears to be in isolated dystonia, cervical dystonia. So you can see here, we've aligned them all to the different subtypes of adaptation and the subsequent washout. The magnitude of the initial error, the rate of the learning, the asymptote that is reached in learning, and then the after effects are almost identical across the control group in red and then the patients with dystonia in blue. And we looked at this in great detail. So for each individual, we plotted their adaptation or washout function and took the metrics from this function to make sure that we weren't missing anything subtle. This is a graph of the learning index, for example, the rate of change at the beginning of each of these periods. And these were very, very similar across the 2 groups. So we then started thinking about exploring this in a genetic group of dystonia. And I think DYT1, a single mutation in TOR1A, are a very beautiful group to try and explore the pathophysiology of dystonia, because we know we are coming from a very pure starting point. They all have the same mutation and a similar, although heterogeneous, movement disorder entails. And this was using the same robot, and this time we were concentrating on a visuomotor adaptation paradigm, where the visual feedback was rotated by 30 degrees. And we also built into this the observation that variability may be very important in dystonia. So very careful work in children from Terence Sanger's lab had shown that variability is increased in dystonia and is a very consistent feature of their motor plans. And then there was very nice papers that were exploring the subtypes of motor variability. And if you actually have variability that is relevant to the task you're learning, at times this can predict a better learning rate. So this is the paper by Morris Smith et al., where task relevant variability correlated with their initial learning rate. So this is now our data, and I'd like us to look at the variability factor VMV, which is maximum velocity. And this was our equivalent of task relevant variability. So the amount of variability there was at the point of maximum velocity during their reach to the target. And hopefully you can appreciate that in controls, this task relevant variability, VMV, and other less relevant variability markers were very little, were not correlated with the rate of adaptation, early adaptation, or the extent of adaptation attained, late adaptation. However, if you then looked at the variability in controls, in dystonia, in DYT1, you can begin to see that this variability is predicting performance outcome, both in adaptation, rate, and the amount achieved, and also the accuracy of the reach late in the trial. So in dystonia, this variability seemed to have an important role. This is a median split of the data, just according to the degree of variability they have. I've put controls in the background in grey, and the dystonics with low variability in orange, and the dystonics with high variability in red. And this is just to exemplify the fact that if you have high variability, you have lower rates of adaptation, both in terms of the initial learning rate and the amount of adaptation you obtain. And so reading into this, what we believed was occurring, that if we had stochastic variability within our system that was clearly not helpful towards learning, such as in the earlier paper I showed you, then this begins to add noise to our teaching signal. And so those with very high variability are likely to have high noise within their system, uncertainty within their system, and are less able to learn adaptation. So on balance, we believe that the evidence suggests that adaptation is not a core mechanism contributing to the pathophysiology of isolated dystonia. And indeed, you can flip this on its head, that actually, if it's intact, then this might be an adjuvant rehabilitative approach to existing pharmacological and surgical therapies. So the next major experimental set that I wanted to review is idling conditioning. Here's a picture of Pavlov and his dogs, and this is a very well characterized paradigm within literature. In humans, the paradigm looks similar to this in that we can repeatedly pair an unconditioned stimulus to a conditioning stimulus in order to generate a conditioned response. And so frequently the unconditioned stimulus is a stimulation to the supraorbital nerve that always without fail elicits an idling, the unconditioned response. And then you repeatedly pair the two stimulus, the unconditioned stimulus and the conditioning stimulus, and with time and repeated pairing, you will start to develop an anticipatory idling to the stimulation called the conditioned response. This is what the EMG data looks like over orbicularis oculi. And you can see early in the paradigm, there is very little contraction. Is no different from baseline before the peripheral nerve stimulation. Whereas when you've had repeated pairings of the auditory tone and the nerve stimulation, you then start to see a conditioned response. And it's pretty unique, I think, conditioning, in that it's observed across the animal kingdom, and partly due to this, we are now very, very confident in terms of the microcircuitry that appears to underlie this phenomenon. So within neural recordings before and then after conditioning has developed, you can see very clear changes in individual cells within the cerebellar circuitry. So you can see the top trace here, where there's zero conditioned response moving through to an established conditioned response. We then see the individual spiking of a Purkinje cell, and you can see the suppression, and then the mean levels across multiple different trials show a clear suppression in the levels of spike activity in Purkinje cells, which is one of the mechanisms thought to underwrite the conditioned response. So we have the potential to have a very unique window into exactly what's happening at a cellular level within the cerebellum, within this paradigm, if we study it in healthy controls and dystonia. Now, I was interested in the fact that we have very mixed results in this paradigm. So early studies with small numbers in focal dystonia had shown impaired conditioning, whereas later studies showed either high levels of conditioning or normal levels of conditioning if tremor wasn't present. So we took advantage of the fact that we could combine all of this data into a mega data set, because all of this data had been collected at the same center with the same paradigm. And we also noted that most of these studies had been run in comparison to a historical control group that was very small, n equals 8. And so we also collected new data in the same lab with the same paradigm, so that we had a viable control group. So we now have 50 patients with isolated dystonia and 50 patients that are and 50 controls. So this is what the range of eye blink conditioning behavior looks like, both in controls and dystonia. And I've plotted them according in a ranked order in terms of those with the lowest conditioning at the front of the graphs towards those with the highest conditioning in the final block at the back of the graph. And I think everyone will appreciate this huge range that we see both in health and in dystonia. Some condition very well from early phases in the paradigm within early block numbers, whereas others show very little conditioning. And we were able to compare this very large group of dystonics to those with no dystonia and show that actually, if we combine all of the subtypes of dystonia, the levels of conditioning are very, very similar. And this group were age matched and sex matched. Now, there's many, many reasons why there's such variability. So in previous literature, it's been shown that both age and sex influence the degree of conditioning. And so younger age and female sex are associated with better levels of conditioning. And we were able to reproduce these findings within this current data set. So they didn't seem to be atypical in any manner to previously published data in healthy controls. So what about the subtypes of dystonia? This is where it really gets interesting. We now have enough data that we can model the covariates age and sex, and we can plot the counterfactuals that are supposedly the result of the subtype of dystonia. And interestingly, one can see that the major groups that we've studied, cervical dystonia, DYT1, DYT6, have very similar rates of conditioning to controls of conditioning. So this is a very preliminary result. So the DYT6 group had only 5 individuals, but all of the other subtypes, focal hand dystonia, cervical dystonia, and DYT1 had very similar levels to control. And similarly, we were unable to reduce the effect in tremor. We found that individuals with dystonia with and without tremor showed very similar levels of conditioning. And so as a final analysis, we calculated the sample size that one should have if we are trying to study this in the future. And because of this huge amount of variability, if, for example, we want to detect a difference in conditioning of 5%, a sample size of 739 people per group is required, which is vastly higher than any of the numbers that we achieved in previous studies. So in summary, I think eye blink conditioning as a proxy for cerebellar function is intact across the subtypes of isolated dystonia, plus or minus hyperfunctioning in DYT6. And this revises early reports that may have erroneously concluded that we have lower rates of conditioning as a feature of focal dystonia. And then it's interesting to stand back and think what this tells us about the pathophysiology. The cerebellum has a very uniform microcircuitry, and some have proposed that this may be indicative of a universal cerebellar transform, that the circuitry is performing the same algorithm on different domains of data in a similar processing format. And so if we buy into this theory, then we could claim that the eye blink conditioning is a global check of cerebellar health, that we check the integrity of the general microcircuitry and plasticity within the cerebellum. However, there's many other viewpoints, for example, multiple functionality, that the same microcircuitry can perform lots of different computations. And so therefore, it may also be true that we're not tapping into this specific functionality at play within the dystonic pathophysiology. So the summary of this experimental data is that many of these fundamental learning algorithms appear to be intact in dystonia. And in a way, this was initially very surprising and seemed at odds with an expanding literature that was heralding the cerebellum as a key node within dystonic pathophysiology. So why might this be the case? And here, I like zooming out a little bit and just looking at this question analytically. What is the role of X and Y? What is the role of the cerebellum in dystonia? And this is actually a very complex question if our role that we're envisaging of the cerebellum and the type of dystonia that we're discussing are loosely defined entities. Furthermore, if we use a probe that is quite indirect, non-specific, or poorly understood, our experiment, to address this question, then very quickly, the answers that we get from multiple experiments will become very difficult to integrate. So we just dwell on each of these in turn. What is the cerebellar role in dystonia? So the first hypothesis could be that dystonia is a cerebellar disorder. Now, I think we already know that there are not two single states of the cerebellum, a binary switch between health and disease, as dystonia is clearly very different to cerebellar degeneration. Therefore, if we have a cerebellar hypothesis for the pathophysiology of dystonia, we know there must be other qualifiers that are needed in order to explain dystonic features. Is it a loss of function, a gain of function? Is it focal? Is it uniform across the cerebellum? If we then move up to the two-node hypothesis that the cerebellum and the basal ganglia are implicated, then a key feature within this is how these two nodes interact. Is one of them compensating? Are they synergistic in their role? However, I think most of us now are happy with the idea that the cerebellum is a network disorder and a wide range of areas have been implicated in pathophysiology. And so the cerebellar role within this immediately becomes very complicated to distinguish. And then some of the subtypes of dystonia quite clearly have environmental factors involved in their pathophysiology. And how these environmental factors interact with the dystonic network is another fascinating area of study. And then the evolution of the symptomatology over time, I think, is another unanswered question. So dystonia often develops even in the genetic subtypes after most of our motor repertoire has already been programmed. Why does this change in motor symptomatology occur? Alternatively, why does it plateau once one has developed dystonia? There seems to be a number of years through which symptoms progress. And then there's a plateau commonly reached. What are the mechanisms behind this? Is this compensation? Is this a new equilibrium, a new set point that the motor system has reached? The other major question is, which dystonia are we talking about? And so there's a very interesting variety of ways in which we can use the word dystonia. Most broadly, we can use it to describe a syndrome, any disorder of posture underwritten by abnormal contractions of the musculature. Or we can use it to describe subtypes of isolated dystonia, where here we are implying that our syndromes that we've labeled descriptively are synonymous with a singular etiology. And there's multiple levels in between this. And here, of course, we're looking at the neuroanatomical level in terms of which dystonias may share the same neuroanatomical substrates. And while we are looking at this question, we need to be very mindful of the experimental techniques that we're using. This is a very simple simulation to try and illustrate some of the points. So if we have an experimental paradigm that we use as a test of cerebellar function, but it's actually only partially dependent on the cerebellum. So 40% cerebellar, 40% basal ganglia, and 20% other. Let's look at different scenarios and what the outcome is in terms of whether the cerebellum is affected in this disease. So here's a 100% cerebellar disease. If we use this experimental paradigm, then yes, we're confident that this is a cerebellar disease. But now we're using it in a disease where it's 100% due to basal ganglia etiology. And again, if we use this experimental paradigm that is split across the anatomical regions, then again, we falsely claim that this is a cerebellar disease. Or if it's a disease of mixed cerebellar and basal ganglia pathology, this test is still positive. And it's not delineating the neuroanatomical substrates for us. This is a very simple point, but in some of the literature that one reviews, I'm not sure that this concept is always fully appreciated. I think experimentally, we're at a really exciting time. So not only is our understanding of the cerebellum hugely increasing in the neuroscience literature, these are beautiful papers that are rewriting the function and the anatomical subdivisions of the cerebellum, and digital. And we also have this expansion of techniques that we can use to try and probe the dystonic network in its entirety. So we already frequently use techniques where we're comparing co-activity of nodes, whereas this is a new technique that was recently published where we're looking at whether the edges between pairs of nodes are fluctuating in a similar manner. So there's new methods emerging all of the time via which we can start to probe this dystonic network. So I'll conclude by using this quote that I think nature seems unaware of our intellectual need for convenience and unity, and often takes delight in complication and diversity. And I think this may certainly be the case when we're trying to explore this question of what the cerebellum may be doing in the pathophysiology of dystonia, I think we're still very far from understanding mechanism at all levels in dystonia, from the genetic mutation through to the final movement disorder. But armed with a knowledge of cerebellar function in health and this amazing toolkit that we're beginning to have to study the cerebellum within the dystonic network, I think we will make very big breakthroughs in hopefully the next few decades. And I strongly believe that this is useful. We have a beautifully dynamic disorder, dystonia, and I do think that most subtypes will be effectively treated with targeted neurophysiotherapy and neural stimulation that can be based on this mechanistic knowledge. So far, we've come very far in improving treatment in a largely empirical manner, and these future breakthroughs in mechanism are likely to yield great therapeutic innovations in the future. So once again, thank you for the invitation to give this talk. I'd like to thank the collaborators and students that helped with this data collection, my academic and clinical mentors, and the grant providers that I have. Many thanks.